last year when Partha wanted me to talk here, he said I should give a tutorial about anatomy and physiology. So I didn't talk much about work in our lab. So this year, but last year I did sort of list about a number of challenges for the visual system, how it solves them. But this year I'm only going to list those challenges and then just about a couple of issues on which we have been working. One is about feature extraction, primarily how the visual system processes the incoming information and codes various features. And also, I'm a classical neurophysiologist and testing hypotheses and ideas experimentally. And only recently we have been doing some computational work. So I'll present some of that. And this coming here has been a great learning experience for me, machine learning and human learning myself from colleagues. And I'll present some of that early work we have been doing. And also other work will be on visual attention, on which we have been doing quite a bit of work on macaques in the 2 decades. Okay. Right. Now, I just list those challenges, which I thought I went to the effort of actually thinking of all possible challenges, so I'll briefly mention them for the sake of students who might be able to take some of these challenges on, how the visual system solves it. One is spatial resolution, because there's 2 opposing demands. That is, one has to collect enough light from so the light could be very dim, so it can collect enough light. But at the same time, if you do that over a larger area, you lose spatial resolution, which is also in the temporal domain when things are very dim and there's a changing intensity, and you need to be able to collect over a large enough time window, but again, then you lose temporal resolution. That's another challenge. And of course, every object has various attributes like form and color and movement and depth, and you have to put all these things together, and we know that there is functional localization in the brain. Different parts of the brain are dealing with different aspects of it, some for color, some for form, some for movement. How do you put all these things together? Okay, and then if you take the eyes, there's another mechanical problem. There's 1 million optic fibers going from the eye to the thalamus and then to the cortex then. And these 1 million optic fibers have to get information about the entire visual scene. So it needs fine discrimination. At the same time, if you have fine discrimination, how do you carry information about every feature? Right, such as color and spatial resolution and everything, and form, movement. How do you manage to do that in those 1 million fibers, right? And the intensity changes a huge factor, right? From scotopic vision, where it's dim, to very bright light. There's a huge variation in light. How do you manage to adapt to that? And how do you make a stable image of the world, because our eyes are constantly moving, and we make about 3 saccades in a second, and how do you, but the world is stable, we don't see it moving, okay? Energy consumption is a huge matter, because our brain is only with 2% of the body weight, but it uses 20% of all energy, right? And it's a highly energy hungry organ, right? How do we manage to do that? So we have to channel the energy to those things which are necessary at any one point. And then if you take any surface, the primary visual cortex or the somatosensory cortex, auditory cortex, if you take a primary visual cortex, there's a topographic map, right? So if you go from one point to the next point in the visual field, the excitation changes. Then you have to have all that on a map, where we also code the features like orientation, color, and code. How do you manage to do that? Okay, and then the sensory overload. There's so much information pouring into a brain all the time, in the visual world, auditory world, and you're still able to focus attention on one. How exactly do that? That's something, okay? That's something I'll be talking about quite a bit. And then when there is damage, the brain manages to deal with it. Okay, now I'm going to talk about a couple of these things. We've just taken some of these challenges, and just what I'm going to be talking about, where we have been working on. Now, most of our work is using an electrode, single electrode like that, or sometimes we do use the arrays. Now started using arrays of electrodes, but this may appear rather simple compared to recording from a large array of hundreds hundreds of, able to record from hundreds of cells like Birkenka showed yesterday. There's a formidable feat what they have done, and also being able to do at different depths. The advantage of this one, on the other hand, is that we have very high temporal resolution. You're only limited by the sampling frequency of a recording system, literally, and the filters that we'll use. Okay, so temporal resolution is very high. Another advantage is that we use these things to train a monkey, and then we record from the monkeys for years. Where you can record from every day, we can go back and record from that electrode. Whereas these things are not possible when you use two-photon imaging. Okay, so there are some advantages to this method. Right, now, a simple model, is the eye, and the information from the eye, the signals are taken to the thalamus and to the primary visual cortex. I'll give you a brief introduction to what they do, because then you'll be able to understand what I'm going to be telling you later. In case you are not familiar with it, there may be some. Does everybody know the visual pathways and all that? Anybody who doesn't know? Everybody knows, okay. I won't spend much time on this. But I just want to show you that that's a cell in the retina responding to light. You can see that it responds particularly to one particular point, and these receptive fields are usually most or less circular receptive fields, right? You can show a bright spot like that, right? A circular spot, or a diffuse light response to some extent, okay? These are little receptive fields which are classically known to be more or less circular, but I'll say more or less, I'll come to that point later, okay? But if you show if you move a bar, unless you do very careful study, you will find that it responds to all orientations. Okay, but that's not the case when you come to the primary visual cortex, right at the back of the brain, where the huge breakthrough was made by David Hubel and Torsten Wiese from Harvard, when they recorded from the primary visual cortex, and they discovered that the primary visual cortex is responsible for orientations. Okay, so for example here. This neuron, a simple cell in the cortex, responds very well to a moving bar of a particular orientation. Just show you very briefly. This is, you can hear that, right? Okay. It responds to a particular orientation. If the orientation is changed, the response is much less. It's David Hubel, I think, plotting the receptive field. There are on regions and off regions. I think we can fast forward that, and I hope it works. Okay. You can fast forward that. I just want to show that a different orientation, the response is much less. That responds very well for that orientation, but it's here. So for this, much less for that orientation. Okay? So this is the orientation selectivity. I'll stop there because I haven't got much time. Right. Now, these receptive fields are supposed to have elongated receptive fields like that. Okay? And they don't respond very well for spots of light, but they respond very well for oriented bars. Now, why am I showing this? Because of all the functions in the brain, this one could argue, arguably this is one of the most intensively studied function of the brain. And Hubel and Wiese discovered one other thing. That is, if you were to record from the cat or the monkey brain and put the electrode in perpendicularly like that, most of the neurons respond to the same orientation. These are the preferred orientation of a particular cell there. But if you go along if you make a penetration obliquely like that, it changes. See, the preferred orientation changes that way, right? Okay. So they made this quite a jump. From this, they concluded that, right, just by recording from single electrodes, that there must be cortical columns going perpendicular to the cortical surface. Nowadays, we have other techniques, like the one that Birkenka showed yesterday, two-photon imaging, or a much easier technique of using optical imaging, where you can actually, when a group of neurons change are active, there are various changes, there are the blood volume changes and the changes in oxidation of the blood. All this can be captured by a camera, and we can actually look at, this is a cat cortex from a lab, where you can actually record the activity like that. Some areas which are more active will show up like that, literally. All right, and then also, that's when the cat has been exposed only to one eye. Only one eye is open, and it shows various orientation. These are ocular dominance columns. Instead of that, you can keep both eyes are open and move a grating a particular orientation, and then you can show up all those orientation columns I showed you. And then you can. This is a pseudo-color map where each of those areas responds best to a particular orientation. Okay. Now, there are two basic challenges that the visual system seems to have solved at the level of the primary visual cortex. One is having a number of selective, the neurons selective for a particular feature. Each neuron is selective for a particular orientation, and they're also selective for the size of the stimulus. We do that with using gratings at different spatial frequencies, and in the monkey particularly, they are selective for the color, the hue of the stimulus, and speed of movement, and the neurons also are selective for stereoscopic depth as well. Okay? This feature selectivity is one thing. Other one is the functional architecture I spoke to you about just now. Now, all this the brain has to achieve, visual cortex has achieved just from the output of this 1 million cells from the retina, right? So you can't have these narrow selectivities in the retinal level itself. We have only 1 million neurons there, whereas in the cortex, right, there are about 300 to 400 times the number of neurons in the visual cortex, right? And these numbers are for the cat, for the monkey is slightly different. You have 1 million neurons for the human, sorry. Okay? So you have this, you can't have narrow selectivities in the retina because you can't have the range of orientation, the range of colors and everything. That's for a color, you know that very well, where you have only 3 receptors, okay, that cover the whole broad spectrum, but you have different peak sensitivities, okay? Now, so how are these things done? Now, we come to some of the models. This is a classical model. This what I told you earlier, this particular property, orientation selectivity, has been so intensively studied by a number of labs. Human research suggested that a cortical cell, simple cell, gets the input from a number of geniculate cells. They've shown only 3 here, but it could be a number of them, but whose individual receptive fields are arranged along a row in visual space. Each of them doesn't have orientation selectivity according to Hubel and Wiesel, and by, if you do tests, unless you do very, very careful tests, you wouldn't see the orientation bias. But they are circular receptive fields. But these the receptive fields of these neurons are arranged along a row in visual space. So when you have a bar of this orientation moving across, all of these are stimulated simultaneously. They all converge here in a cortical cell, you get a good response. If it is a bar of this orientation along here and it moves across, it doesn't do that, so it doesn't get a good response. But this has been intensively studied because as if, and I just give the names of some of the labs here who might have it. Hubel and Wiesel also had a suggestion of hierarchy level in the cortex. Within the cortex, what happens, different types, different cell types. Right? There have been alternative schemes suggested, and some people have been in both camps, 1 foot here, 1 foot there, but I was trying to classify them to some extent, right? American Cock is also here, I don't know, he is also 1 foot here probably. Okay. So there have been a number of labs who have been working on this. But over the years, the Hubel and Wiesel's model has kind of stood the test of time in some sense, but in other ways it hasn't. I'm not going to go into all of this in detail. There are various challenges to that model, right? So one has to make some ad hoc changes to the model. But finally, there are some observations which have been rather difficult for the model particularly. That's why here I have chosen some of them. But one of the things I'm going to point out now, the fact that orientation selectivity doesn't arise de novo for the first time in the cortex. Now, I believe that the biological evolution is biology is a hacker. It uses whatever is there to make the best use of it, to make the best use in a particular niche. Now, orientation bias are already present in the retina. we saw this quite a while ago. So this is neuron in the lateral geniculate nucleus. That's in the thalamus, that's the retina, thalamus, and the primary visual cortex, right? This is the one orientation. There's a good response. The other orientation, the response is much less. This has been seen now by a number of people in almost every species you take, mouse, ferret, tree shrew, monkey, cat. You do find this orientation biases in the retina and the geniculate. Now, this is only a bias. It's not a very sharp orientation selectivity that you see in the cortex. In the cortex neurons will respond only for range, say about 20 or 30 degrees range. But in the geniculate retina, they respond to all orientations usually. There's never bias to one orientation, okay? Right. You can do it with gratings, with bars here, done by Levick and Thybus. In the cat, you find that as you change the orientation of the grating, the response changes, okay? And these are the polar diagrams showing the response in different orientations. So they're sort of elliptical here. They're not circular. If there is no orientation selectivity, they'll be circular, right? Okay, it's just again with gratings, you're showing that low spatial frequency, there is not much of a bias. It's all in the medium and high spatial frequency. It's a point we can discuss later if you're interested. Now, if you have such a biased input, I said biology is a hacker, evolution is a hacker. So all you need to do, and we know this happens, that all you need to do is that if you have a biased input like that, you create some non-specific inhibition even. It's a kind of a tip of the iceberg. That's all you do, and we know that this kind of non-specific inhibition does occur in the cortex when the signal comes to the cortex. And then, of course, what you have is a response like that. And of course, the various mechanisms by which this response can be amplified. The various intracortical mechanisms are there, and there's yet another mechanism which I'll come to you later. There's cross-orientation inhibition, which is inhibition from other orientations, right, which actually can increase its orientation selectivity, right? And finally, and we have done some modeling of that particular scheme using input as gratings and controlling that with a receptive field here. There's a slightly biased LGN cell, and you can even have another LGN cell which is unoriented, which doesn't have orientation, and inhibition coming from that, because of course all fibers from the geniculate to cortex excitatory to the inhibitory interneuron in the cortex, you can get that. Or you can also have the other way around, the inhibitory interneuron, the neuron giving inhibition could be biased, other one could be also biased, or it can even be circular. You end up in nicely oriented receptive fields. We have simulated that very well, and it seems to match the results we get. So I'm going to taking you through the history from human weasels to right up to what we do now, which is still unpublished. So I'll take you through the whole lot in a way. Now, you go by the classical human weasels excitatory model here. Essentially what they're saying is retina, receptive fields are circular, a number of LGN cells like that, they all converge onto one cell, so this becomes kind of an elongated field, and you get orientation selectivity. This has a few problems. I just pick on a few, right? One is that if you look at the central excitatory region of a cortical cell, do that with the intracellular recording, which you can do when you can look at just say excitatory or the inhibitory postsynaptic potentials. When you look at that, the excitatory discharge region is not elongated. It's actually pretty almost circular, or only a small bias, more or less the sort of bias that you would see in the geniculate, single geniculate cells. So that is one problem. And then if you do, this is quite a formidable experiment that people did, that Brian Cleland, Barry Lee, Nora Kreuzfeld did that. They managed to record from a cortical cell and a retinal cell simultaneously, the retinas, and managed to find in the retina the cells which project to cortex. And they found that if they found a match, they found a 90% of a cortical cells, almost 90% on average, but 90% of the spikes arise from a single retinal cell. So that is quite a challenge to the Hubbell weasels model. It arises from one retinal cell in the cat. Yeah. No. Why are they geniculate? It's recording from the cortex and the retina, right? No. Through the geniculate. the geniculate synapse, and I'll come to what happened to the geniculate in a moment. So if you record, ultimately the signals have to come from the retina. So you can go and you can find the receptor for the cortical cell, and you can put the electrode intraocularly, and you can go and find actual. It's quite a difficult experiment, but they managed to do that. So then they do, when you find that most of the cortical cell spikes comes from a single retinal cell. Okay. Now, yeah.
How was that shown? Was that done by cross-correlating the retinal cell and the cortical cell?
Yes. Yeah. I can give you the paper somewhere here. I can give you that.
So how can you be sure that there is no other?
There could be, but what I mean is, from no cell, if either you get a high degree of correlation or you get a very low degree of correlation from the retina. If you go to the retinal area and poke into different cells, you find that for one cell you get a high degree of correlation, all the others will be very low.
Confusion is not a trivial thing to prove, but we can talk
Yeah,
about
okay, all right. And then, but on the other hand, Tanaka, Keiji Tanaka, in Japan, they recorded from a cortical cell, a geniculate cell, and they came up with the result that 10 geniculate cells on average convert onto a single cortical cell. So there is convergence. And there's also anatomical evidence showing there is probably convergence of the order. It differs for different cell types. I'm talking on average for 10 cells. Right? So what's happening? Retina to cortex 1 to 1, geniculate to cortex 10 to 1. Okay. Now, here, the clue is this. A single retinal cell diverges to about 10 cells on average. Single retinal cell diverges to 10. So these 10 geniculate cells will have identical receptive fields. Okay? Okay, this is again different for different cell types. For X and, there are different cell types in the retina, X and Y, but I'm talking generally. This comes from one retinal cell. Now, Hebbian plasticity operating on this pattern can solve a conundrum, right? For those of you who may not know what Hebbian plasticity is, that Hebb postulated that once one cell excites another cell here, and if it repeatedly and consistently takes part in firing this particular cell here at one synapse here, this synapse will become facilitated. Okay, so and it's depending upon only for that synapse, not for another synapse. This is basic Hebbian plasticity. Many of you may know all this anyway. So anyway, you heard also from Peter Dayan yesterday. Okay, now I have a graduate student, Harold Lloyd. He's very much interested in maths. Along with some colleagues at the engineering department, Katie Davey and Tony Burkett, we did some computations here. Then, so he had a simple model where you have, as I said, a retinal cell projecting to about 9, I think, or 10, 9 LGN cells, and the LGN cells show this kind of a single V1 cell can get in from initially before the plasticity. There's a protuberance. Quite a lot. We know that LGN projections go much more or a much very large area, and the early period, the few weeks, there is quite a lot of pruning of these things. But there is obviously some Hebbian plasticity going on. Now you know, as I told you earlier, these 10 geniculate cells will be highly synchronized among each other because they get from one retinal cell. So the Hebbian rules will, which is what is modeled in this one. This is before plasticity, right? And you have a very simple linear rule. And after that, during plasticity, depending upon the correlation between the V1 neurons and between the LGN neurons, then finally this kind of thing is likely to evolve. Okay. So, but here is, so that is easy to follow, that how Hebbian rules will channel all the input from the geniculate, which ultimately arising from one retinal cell onto one cortical cell. Okay? Now, this also is a general solution for the functional architecture of orientation ocular dominance columns. Okay? I'll take an analogy with color. in color, you can't possibly have very sharply tuned pigments in the retina. Of course, behaviorally, we can distinguish hues of very narrow, differing only in a few wavelengths. And the cortex, you do find cells which are very narrowly tuned. And even tuned to things like purple, which is not in the spectrum. Right? But you can't have 100 different pigments in the retina. Then each cone, as you know, there are 3 cone types, red, green, and blue, you call them, long wavelength, medium wavelength, and the short wavelength. And what they do is that what we have is, you have these 3 pigments, 3 cone types, which are very broadly tuned, but the peaks are different. And the cortex compares these ratios of these 3 and finally elaborates the whole spectrum. What we're suggesting is that is the same, not just for, it's the same for all parameters. Any parameter which is coded in the periphery, and the cortex elaborates from that all the range of whether it's color or the orientations, the movement, it uses certain simple Cartesian coordinates. So any orientation can be coded into Cartesian coordinates or 2 or 3 limited number. And from that you can generate the whole thing. So if cortex is actually depending on these orientation biases, because when we found these orientation biases, when we spoke to David Hubel, he was saying, oh, these are only biases. Cortex is a very sharp orientation. But my answer is that the same in color. Color also, you find them only very broadly tuned. Okay? So in orientation, you can also imagine that there are a few orientations, right? And the primary orientation, let's say here, vertical and horizontal, you can create the obliques. Of course, there is a problem with breaking the symmetry. That's a different issue. I can talk about it later. Now, okay, what about so it's only biases. We find the biases, these are only biases, that's fine. What about the limited number of channels? Right, in the color you have 3 channels. In the orientation domain also, it seems to be that this is a work by Levick and Thibos, and now it has been shown also at the geniculate level, and we have seen it a number of different ways, that at any particular location, the retina, most of the biases seem to be, the preferred orientation seem to be along this radial orientation. Or sometimes it's orthogonal, right? But mostly the radial is dominating. You find the same thing at the geniculate level too. And also in the human vision also, you find a bias actually for radial orientation as you go away from the fovea. But close to the fovea seems to be vertical and horizontal, okay? So you could create this, what I've shown here, that you have initially, say, the 2 biases, vertical and horizontal, and before plasticity you don't have that. But going to the plasticity phase, where the Hebbian principles come in, and you have this pruning of this inputs to the cortical cells, you would find the cells in between might be getting oblique orientations. In fact, Errol has, that's the wrong one. Okay, here, just I want, these are you can pick on any particular cortical cell here. Wait. Okay. All right. What you see at the top is the retinal receptive field, is the retinal input beforehand, and this is what happens after the Hebbian plasticity. So essentially, you find that this kind of diffuse projection from various retinal cells finally come to just the 2 of them there, and if you take with the whole population here, you find 2 things. One is that the overall orientation selectivity is a circular variance, which means that one means there is no orientation bias at all. And here, orientation selectivity goes up here. Overall, it comes down. Not only that, you start creating these other orientations in between. This is a very simple rate-based model. There's nothing more to it. Right? We haven't introduced anything else here in this one, such as intracortical inhibition or other cortical mechanisms. Okay. Now, I'm just showing you only this. I've just spoken to you about orientation selectivity, but it's true for, you can apply that to other cortical properties we have, like ocular dominance columns and selectivities for other parameters as well. So if you take here, it's got a particular set of fibers coming from the geniculate, which arise from one retinal cell. It would have its right eye or left eye, its on center or off center, at the same time is one orientation or the other. So in between that, you will create these orientation columns. So the creation of the orientation columns is an inevitable feature of the way orientation selectivity itself is set up. Okay. This is the more or less the same thing I showed you earlier. So you find that without any other additional mechanisms such as intracortical inhibition, is simply based upon the excitatory input alone. Once you give this kind of orientation bias and these two Cartesian coordinates, here three of them actually, vertical, horizontal and radial, and you create all this additional orientations here you find in between, and the overall orientation selectivity goes up. This is before circular variance. Before that, that is here, the orientation bias is only low, and it increases quite sharp selectivity later on. Okay. And this shows, and also shows the percentage of the retinal, how many retinal cells contributes to a single cortical cell, the percentage before plasticity, and after that it becomes only almost single cell, as contribution comes from a single cell. Okay. Now, we did a few other tests of this model. If this is true, then if you look at the afferents coming to a particular cortical column, you should find a match between the preferred orientation of the cortical cell and the preferred orientation of the afferents that are coming in there. Okay? Or the cortex you may say the cortex may ignore all of this and use this Huber-Wiesel's model, right, to create orientation selectivity. Right? It's done by my students here and postdoc. Jay Kishan is over here. Oh, not here. Suddenly he disappeared. Okay. What we have done is that we did optical imaging, as I said before, you can create the orientation columns, and then we applied cyanic acid, which actually destroys the cells, leaving only the afferent fibers there. Okay. Or we gave muscimol, which is an inhibitory, which inhibits the activity of cells, and then you can record from the afferent fibers coming in. Okay. So we used very, very fine-tipped electrodes, which you can record from afferent fibers. And we found that this is the sharp orientation selectivity of a cortical cell before we applied kyanine at a particular place. And once we applied, this is in the case of muscimol. Muscimol suppresses all activity of cells, leaving alone only the afferent fibers. This is the orientation bias of a fiber. You see that the same preferred orientation as a cell. These are three different sites where again you find that in one case the orientation selectivity of the cortical cell, and after suppression of the actual activity of the cells, when you record from the fibers coming in, they more or less match the orientation of that, right? This is again afferent orientation preference and the cortical orientation preference. They match quite fairly well. They shouldn't match everywhere because you also create other orientations at some points. Okay. Now, another prediction we tested was the same point we tested in a different way in macaques. That when we do already know about optical imaging. This is the classical optical imaging you do, because when you do optical imaging, you do a lot of filtering, high pass filtering, low pass. You can use a band pass filter because there is quite a lot of signals coming in, and the effective signal often, when you want to match the activity, when you want to match the optical imaging activity with the activity of single cells, you're looking at the postsynaptic cells. It's not the afferent activity that's coming in, right? But the majority of the signal you get in optical imaging is actually related to the activity of the presynaptic activity, synaptic activity, and the dendritic activity is not the postsynaptic spikes. So it's only when you use a bandpass filter and in fact cut out most of the low spatial frequency, you are able to see this beautiful orientation map. And there you can put a cell there, you can put an electrode there and record from the cell the spikes, and it matches fairly well. Okay? But what we decided to do was don't do any filtering. Look at the raw signal. What is coming into the cortex? If you look at the raw signal, it's dominated by one orientation. And that orientation is the radial orientation. All you need to do is that don't apply any filtering. No Jassy filtering. Just look at the raw signal, right? And you find it's the radial orientation, right? Okay. Now, this is for other monkeys. You find the same story because I want to go to the attention story soon. And to quantify that, we took a number of little pixels here, 30 by 30 pixel. I looked at the average vector there, and then and also we actually recorded from the electrode, and so that we know the receptive field location. We can get the radial, and then we can get the actual radial angle for that particular topographical location. So we had to do recordings as well, single cell recordings. And when you did that, and we find that the veridical map or the unfiltered map, there's not much difference between the vector, the average vector in the optical imaging, and the radial orientation of the topographical orientation. But the filtered map, it becomes different. That's when you're looking at probably more the spike activity. Okay. Now, quickly want to go on to the attention bit. Now change your attention to something else. One of the common things we need to do is looking for a particular object in a cluttered world, looking for a key in a cluttered desk, or looking for a car. You have forgotten where the car was in the car park, and say your car is over there, and we forgotten where it was. We still remember it's a red car. We know the shape. We know what model it is. Initially, we tend to do it because there are various psychophysical experiments which show what we tend to do in these situations. We do what's called a guided search. Initially, there's a parallel stage. We pick up all the red cars, right? Everything else you blur out in some way, not physically, but cognitively, and then you do a serial search among them, right? Like that. Okay. So for that, there are two questions. You need to create a saliency map initially of all those important red cars, and then you do a serial search in that. Okay. Now, how is that done? It seems that the intraparietal cortex, which is a part of the posterior parietal cortex, which Mruganga talked about, it seems to be involved in top-down attention. But does it have the equipment to do this two-stage of guided search? Okay. Now, what we did was we record simultaneously from LIP, that's the posterior parietal cortex, and MT, which is an early visual area. So signals come to V1, as Mruganga showed yesterday. They go to the dorsal stream here, and the ventral stream. Dorsal stream is more concerned with spatial issues, where things are, but it does have some crude feature selectivity as well. Whereas the ventral region is concerned more with what the object is and our perception, and there seems to be some sort of a top-down mechanism which probably spotlights certain areas, okay, on the map. Okay. Now, the thing is, there is a sensory input has to come into LIP via V1 and MT, that's one thing, and it just create a saliency priority map in LIP. There must be coarse feature detectors which are doing that. And there must be a serial feedback going back to MT, okay? Is it happening there? So we managed to do simultaneous recording from LIP and MT during an attention task. The monkey, it's quite a complex attention task, it takes 2 years to train the monkey. And then we had to record from both areas simultaneously and find the topography of the corresponding regions and the matching receptive fields as well in terms of orientation selectivity. And we did that. Yuri, who is now in Wisconsin, and Ivan, who came from Moscow. And this is the task. I won't go into too much detail, but just to show that the monkey is presented with, has to match two gratings which are presented with a delay between them. Okay? There could be a match case in which it has to match both where the stimulus is, where the second grating is, and also with the same orientation or not. This is a match case here. Or it could be a non-match where the orientation is different, or the location could be different, or both the location or orientation could be different. All these are non-match cases. The monkey has to respond differently to the two things. Okay? Now, we find here that that is increased with the first grating and second grating, and these are, when they are matched, we find a good response there. It's called attentional modulation, even though the stimulus is exactly the same here and here. Once the monkey pays attention, it's much greater. Yeah. All right. This is an MT, this is the LIP. And earlier we have shown that this LIP activity here going up, ramping up, seems to derive this increased activity in MT. Okay, this kind of a top-down modulation happening from LIP, which seems to be happening due to synchronized oscillations in LIP, which is actually deriving the MT neurons, right? It's like the neurons sync together in LIP and they derive these MT neurons at the same location. It's actually as if you want to push a car, if push start a car, four or five people pushing it have to do this simultaneously, right? You can't do it independently. It's similar to that. Okay. Now we found in LIP two groups of cells, what I want to talk about briefly. One group, they're showing attentional modulation like I've shown before. The second grating, the response is much greater than the first, but they don't have any feature selectivity. But the second group of cells, which doesn't have any attentional modulation, the response is the same, but they show great feature selectivity. This is an extreme case, of course. They grow great feature selectivity. It's as if either you have a high degree of feature selectivity, but no attention enhancement, or you have a high attention enhancement, but not much feature selectivity, right? This is more or less precisely what one would. This is largely analyzed by Katia here on the data I collected earlier with Ivan and Yuri Salman. Now, to see here, the red points are the red dots are the ones where the cells are the ones which show attention enhancement, but they don't tend to have much orientated selectivity, right? But the other cells which do not show attention enhancement tend to have more and more orientated selectivity. Okay. Now we also find that this ramping up that you see here before the second stimulus, which almost ends up in the increased attention enhancement, in the attention enhancements plus cells, right? It doesn't have much activity early on, but the other group of cells seems to have higher activity here. This is the ramping during delay index. This ramping index is very high here, but not here. But here, what we find is that, I haven't got the slide here, but you have significant activity just immediately after that first half, okay? And we also find that these attention enhanced cells also show a lot of local synchrony. This is synchrony which probably helps the empty cells to act, empty cells to be facilitated, okay? But whereas the feature selected cells do not show such local synchrony, right? Later on, but show it in the early delay period. It's as if these cells are keeping the memory somehow and passing it on to the AE plus cells. But because initially you need to select those red cars, right? You need to saliency map based on a feature selection, based upon features. But the next stage, you just want to look at the location carefully, right? You don't need any feature selectivity. And that's what probably these other cells don't have much feature selectivity. Okay, this is again, it's a complex step. I don't have time to go into that. If we're looking at degree of coherence between LIP and MT, you find then the coherence seem to be in different bands, right? For the feature selectivity seems to be in higher bands, higher gamma frequencies, whereas and that is more in the early part of the delay, whereas for the attention enhancement cells, it's somewhat the lower frequencies in a beta and low gamma. That is the later part of the delay. Okay. All right. Now this is just a model which captures that, where if you have that's the target. Initially, if that is the target, you pick up these locations and then you do a further spatial serial search along those. This is something we haven't simulated, but probably, hopefully that I can collaborate with people. I mean, I'm not a computational expert. I just have one student starting on it now, so it'll be good to simulate that. Okay, now this is just summing up. So you have at the level of the primary visual coordinate connectivity scheme, which uses the asymmetries that are already there in the retina. So as I said, evolution is a hacker. It uses these biases. In fact, these biases created by the way the retina grows. It grows from a center like that, and the dendritic fields get elongated as it grows. So the biases are already there. Okay, and the way the biases are combined can produce the whole functional architecture of the cortex as well. Right, and the next one I talked to you about is how the parietal cortex seems to be having these two stages that is necessary. But of course, in between that is what a hidden layer, which we don't know, because that needs to take into account what the executive functions are, right from the prefrontal cortex. That needs to take the command, what it needs to do. There needs to be hidden layer. I only spoke about the two types of cells, the feature selective cells and the attention cells, but in between there must be a hidden layer which somehow incorporates the higher executive commands. Right, now briefly I would like to acknowledge a number of people who have done most of this work. Katya, she joined me from Moscow, has been with me at postdoc a number of years. Yuri, he went to Princeton and now in Wisconsin. Moji is from Iran, a graduate student. Ivan, a colleague from Moscow. Yamini is a graduate student who's been doing some of the work I told you with optical imaging. Jay Kishan has been involved in a number of work, a number of experiments, and Errol and Levin have helped me with the simulations. They've done the simulations, and Ulf has been a long-term collaborator from Bochum. Thank you. Yep.
One is color. Is it maintained as separate streams or combined as you march up?
Yeah.
And I'll ask the other question at the same time.
Yeah.
In the so-called deep learning models, people are using hundreds, thousands of layers.
I mean, color is somewhat.
What's your sort of feeling about that?
In this middle layer, the geniculate, something else happens with color. That is, it forms two opponent channels, right? There are essentially three, red, green, and blue, and you find the red-green channel and the blue-yellow one. Blue-yellow has the short wavelength input, and it combines the red and the long and the medium wavelength pulse inhibition, and so you create two channels essentially, two kind of Cartesian coordinates, and from that, in the cortex, when you go to the cortex, you find the whole range of hue preferences, but not just these two major preferences that you find in the geniculate, and you also even find purple, which is a mixture of red and pardon? For the cortical areas, I mean, in V4, in fact, there is a different transformation happens that we know because there's what's called color constancy, right? Because that's something that's been shown by Sami Zeki and others. One remarkable thing that has happened between V1 and V4, which is originally named the color area, is that depending upon illumination, we still the illumination may change to some extent, right? For example, in midday it's very bluish, and in the evening it's more reddish, or when you have fluorescent light or the usual globe, which is highly reddish, the colors don't change. This kind of color constancies seem to happen there, but the same the range of color hue preferences that you still see in V4. That's maintained. Yeah, that goes beyond. I was only talking up to the primary visual cortex, right, where you code at the level of orientations and colors, simple basic features. But when you come to complex features, object recognition, that is a very difficult beast. I don't know how exact. But we do find cells which are tuned to very complex objects, right? And how that created at the neural level is still probably.
So my question is, before you get to the object recognition, you probably haven't passed through 100 synapses.
I don't know whether we need to pass through 100 synapses, but I mean, we don't have 100 synapses.
The deep learning network that Ricardo has made, he seems to model the multiple layers on the hierarchy of the ventral stream from V1 to
Yeah.
V2 to V4 to
TE,
various
TE.
layers of IT. His work has emphasized
That-
IT itself
that-
is
that-
fractionated into 4 or 5 different layers. So still we are talking 6 or 7 layers.
6 or 7 layers, right? Not 100.
Those layers in a deep learning network is just still one connectivity. Whereas in the cortex, there's probably many more. Interlaminar, then feed forward, then interlaminar, feed forward.
See-
So it's much more.
see-
I just want to
Yeah.
point this out as an obvious disconnect between the two. When
Yeah.
it's something to learn from
Yeah.
comparing these architectures.
Yeah. Like you mentioned on the first day, on the one hand, machine learning has done things very effectively, certain things, but we don't probably do it that way because. On the other hand, there are certain biological constraints. You talked the issue about sparse things. What's the point of having things which are correlated because it's redundant in a way? All the neurons seem to be doing the same way. But I think there are certain biological constraints you have to take into account, and that probably prevents you from sometimes having sparsing, for example, to do this kind of synchronous oscillation is necessary for one area influencing another area. So, but again, within an area, as Mriganka pointed out, it's better to have decorrelation.
I can have a quick question
Yeah.
before we end. In parietal cortex,
Yeah.
you show 2 kinds of neurons. One that are visually driven and hence orientation selective, and the others that are, and I did not understand that part. Are they visually driven?
Oh yeah, very much.
But they are highly attention modulated.
Highly attention modulated, very.
What is the location of whenever you have multiple cell types in an
Yeah,
area, you
yeah.
want to know.
Yeah.
You assume they are unique in terms of their inputs, perhaps their output in terms of their local system.
Yeah, I mean, is awake behaving monkey? I don't know which layers they are morphologically. I don't know, but what I can tell you is that they seem to be together, clustered. There seems to be a certain amount of clustering of these neurons. So they could be in different layers. That's all I can tell you. But one thing we are trying to do at the moment is doing spike field coherence between MT and LIP. Spike in one and the LFP in the other one, to be able to get the direction.
But you obviously believe that they are part of the same computational process in terms of your model.
Yeah.
One is for broad scale,
Parallel.
paraphrase, sort of sampling, and the other is for fine
Focus spatial attention.
Would you consider the possibility, it's a little bit unusual to have that neuron. Would you consider the possibility that they actually have very different projections? Because the parietal cortex projects to many different areas, including to the striatum and to the frontal cortex and back to the visual cortex as well as to the thalamus, and these are probably different cells.
I agree that it could have other functions, but I think before the second attention cells, they need some feature, some kind of direction based upon feature. Otherwise, they can't choose their locations. So,
This will continue. I think we need to thank you very much. Then we will need a sharp.